Okay, hello everyone. I'm Tom Lichtefeld at Johns Hopkins School of Public Health, and today I'm going to talk to you about my first rotation on 3 projects, one in the Center for Imaging Science, one in the Center for Alternatives to Animal Testing, and one on a web application called Metascopics. The first project in the Center for Imaging Science was with Dr. Jane and Dr. Jedinak. We were tracking tuberculosis development through non-invasive imaging. This project starts out with a low-dose aerosol infection, tuberculosis, and then anti-tuberculosis therapy. There were actually 3 different therapies being tested, and during this period we had imaging indicated by the blue arrows, and the idea was to track how granulomas, which are lesions caused by tuberculosis, change over time during therapy. So how do we do this? Well, there are 3 different imaging modalities. There's first computed tomography. The computed tomography is going to show us where the bones are. This is primarily important because when we're tracking a biomarker, which we'll talk about later, we want to make sure that we're only looking at it in tissue and we're not getting noise from bones and other things that can be detected by computed tomography. So to track biomarker, we use positron emission tomography and single photon emission computed tomography. The biomarker we used was one that was specific for glucose, and here you can see in the images what are probably different granulomas, but also the heart is positive for glucose activity. And in the computed tomography images, you can see the rib cage and the spine. And basically the idea is that you want to segment out the lungs and also the heart. So what do we do here? Well, first we have a template lung, and we want to match each of our mouse lungs to that template. We have a positive segmentation, which is aided by the user, and essentially you're identifying the lungs in an individual mouse where our biomarker is active. And then we have a negative segmentation where we have bones, the spine and the rib cage, and we're using both of those positive and negative segmentations with some segmentation algorithms and registration algorithms to register those segmentations onto a template lung. So we get the template lung is important because we would like to compare our mice, and it doesn't make any sense to compare mice with different lung morphologies. We would like to match all of those different lungs onto one template lung, and we can see why in a moment. So once we've done that registration, we can look at different voxels, and we can see what the intensity for our biomarker is in each of those voxels. We can see that in a control mouse, the intensity is going to be higher near the heart, and that there's going to be quite a bit of variability near the heart. What that means is that if we end up getting signal near the heart, we end up having to have a whole lot more signal to be sure that there's a granuloma there. So this is used. This variance is used in a normalization procedure, and what we end up getting out of it, you can see down here in a subject image, granulomas. So these are images produced by Dr. Bruno Jedinak, and essentially what I was working with was putting the workflow together to put together different imaging software, MIPAP and Amira, with the algorithms developed in the Center for Imaging Science using MATLAB and C++. So what else do we get out of this process? Well, we figure out where the granulomas are in the lungs. So we can, usually the lungs are divided up into different sections, and we can figure out how the granulomas are changing over time in those regions. And that can tell us about how effective our therapy is being. You know, maybe it's more effective in different regions than others, what regions are usually affected by tuberculosis, and also it gives us a quantification over time of the biomarker activity. And so, you know, that time series can be used to perhaps predict relapse later on. So in my second project at the Center for Alternatives to Animal Testing, we were doing skin sensitization potency. The local lymph node murine assay is a surrogate assay for skin sensitization. It's a assay used in animals. Essentially, you put a little bit of your chemical on an animal ear, and you see whether or not they develop tumors. It's kind of a, you know, from an animal care perspective, it's not such a nice test to be doing, and so it would be great if we could figure out some in vitro assays to take the place of that test. And so this is a Bayesian network developed by Joanna Jaworska. You can see the local lymph node murine assay here, and it's you know, it's divided into 4 different categories: non-sensitizers, weak, moderate, and strong. and you can see that different variables are dependent on the value for local lymph node mirroring assay. This is how Bayesian networks tend to work, that the target variable is causing different values in the predicting variables. The blue variables here are in vitro assays. I won't get into them too much, but essentially they're assays for NRF2 and direct peptide reactivity assays. And then we also have chemical property variables, and then we have an expert system, which introduces some problems that I'm not going to get into in cross-validation. Essentially, if this expert system is trained on chemicals that we're creating our supervised model for, you can have some overfitting problems. So what are integrated testing strategies? This is the big idea. This is what we want to create. Essentially, what we have is two parts to integrated testing strategy. First, we have what is ideally a perfect supervised learning model. We want a model that takes a whole bunch of data. In this case, we're taking data for rain tomorrow, and we're trying to say we want to create a model that perfectly predicts that, or at least predicts it pretty well. In this case, our Bayesian network actually has some arrows in the wrong direction. The rain tomorrow should be pointing towards cloudy and raining nearby. But the point is, a Bayesian network can handle interdependencies between the predicting variables. So what do we get out of that? Well, we get joint probability tables, but more importantly for the process I'm about to tell you about, Bayesian networks and several other supervised learning models can do predictions with unknowns. So this brings us to the second part of integrated testing strategies, which is testing. So testing evaluation. What's the big idea here? Well, after we've got a supervised learning model that can do a really good job of predicting skin sensitization, we might want to say, well, now I've got a new chemical. Which test should I do first? Well, if all the tests cost the same amount, the test you should do first is the one that's going to tell you the most about whether or not the chemical is toxic. But what does that really mean? Tell you the most about whether or not a chemical is toxic. Well, what we did was take a greedy approach, and the idea was to take tests that had the greatest divergence in target values. So if our target has 4 different values, non-sensitizer all the way up to extreme sensitizer, the ideal test divides our domain space of chemicals perfectly into categories for that target. So if we had a test that did that, our most valuable next test would indeed be that perfectly predicting test. And then we wouldn't have to do any tests after that. We would already know with extremely high confidence what kind of a sensitizer our chemical was, and we would also be able to see that doing more tests wouldn't improve our prediction at all. And so the great idea here is that if we can identify these most valuable next tests and do it repeatedly, we create a decision tree-like structure, and we can hopefully dramatically reduce the amount of testing we have to do and save a lot of money. And so I told you that you can use different kinds of supervised learning models to do this. We tried 2 different models. We tried a random forest approach with our data, and we tried the Bayesian network approach. We thought that maybe there would be a problem with the times model, and so we didn't use that for the random forest, but that's another story. Essentially what you see down here is cross-validation and balanced accuracy. So before you've done any tests for a new chemical, both of these models are going to just predict the most prevalent categorization. And so what that ends up getting is a 25% balanced accuracy, because each model is going to be 100% accurate for whichever endpoint is the most prevalent, and 0% accurate for the other 3 endpoints. You can then see after we've done one test, which would be the most valuable test globally speaking, that both of these models have a substantial improvement in accuracy. And then as we do more tests, accuracy goes up for a while, particularly in the random forest, but then it seems to level out. And so that would indicate to us that our algorithm seems to be doing a good job of providing the most valuable next test, and that at some point we could probably stop doing testing. I think that this model needs to be improved a little bit. Essentially what we would like to do is have good ideas of what the confidence level is at each point in testing, and we would like to be able to say how much improvement in accuracy we would expect to see for more testing. So that we would be able to create a well-defined cutoff for where to say you should no longer do any more testing. It won't be helpful. So finally, I worked on a web application during this time with a student in molecular immunology and microbiology, Sabrina Nolan. And the idea behind this web application is to combine life science data with machine learning algorithms. Sabrina Nolan's project involved image analysis, so that's the first stuff that we developed for Metascopics. The 3 motivations behind Metascopics is to bring reproducibility, applicability, and visibility to algorithms we see in life science applications. So reproducibility is a really big one. A lot of the time you see a new algorithm in a life science application, and you don't really know if the algorithm has been applied correctly. Overfitting can be a really big problem for machine learning algorithms, but there are other various problems that occur as well. And so what you would like to be able to do is to take someone's data and take the algorithm they used and run the application exactly the way they ran it. If you could do that, you would be able to look at their workflow and say whether or not there were problems in it. So that's the idea behind Metascopics. We provide an online link where you can give this link to people. They can look at your workflow, they can look at your data, and they can see how your data runs through. Additionally, we provide greater applicability for new algorithms, because now if someone can see how your great image analysis algorithm works and how it does a really good job of identifying whatever it is you're looking for in cells or parasites or what have you, they can take your algorithm and directly apply it in their own projects and directly apply it to their own data once they've uploaded their data onto the web application. And finally, we increase visibility, because now rather than having all that data stored in cloud storage or on personal storage, you have it in a place where people can go look at it and they can see how it works into a workflow. And they can see how that works into your algorithm. And now you can just give your link to coworkers. You can give that link to friends even, or family, and you can have people take a look at it. Of course, we want to provide privacy for data that wasn't ready to be published yet or wasn't ready to be seen. So let's take a look at this actual application. We're hoping to get a publication in September of 2014 with this work, and these are just going to be example data. So in this first, we had 3 algorithms developed. In the first algorithm, Sabrina Nolan wanted to know whether or not the parasite she was looking at, which is Toxoplasma, it's a parasite associated with cats, whether or not the parasite was recruiting the Golgi body. So we thought about it for a while, and we thought maybe a way you could do that would be find the centroid of the Golgi body, and then find the distance between that centroid to the closest surface on the parasitic vacuole. There are some drawbacks to this approach that I don't have time to get into, but you can see that in the data, at an early time point, we have a relatively large distance between the surface and the centroid of the Golgi body and the parasite vacuole. And as time progresses, that distance reduces. You can see an image of this at an early time point and then at a later time point. Another approach we had to this problem was identifying for both Toxoplasma and Neospora, which are 2 different parasites, whether or not they're recruiting the endoplasmic reticulum, which is a larger organelle. So to do this, we again used immunohistochemistry, and what we did was we wrote an algorithm where the user would draw and identify the parasite vacuole, the surface of the parasite vacuole. The algorithm would then create shells around that vacuole, shells of 1 pixel diameter, and then we would count the intensity. We would sum up the intensity in each of those shells. So the total sum in all of the shells would be the cell host endoplasmic reticulum. Within one shell divided by the complete sum, we would get the percent of the ER within that shell. Of course, we might want to normalize for the shell area or something like that, but even just using this approach, we see that at early time points, the ER is going to be pretty well distributed, both away from the parasitic vacuole and close to it, and that at later time points, the ER tends to be more focused near the parasitic vacuole. And this is not a cumulative probability, this is probability at each shell. Again, these graphs are created by Metascopics, and the images you're seeing here are really pre-alpha Metascopics, but hopefully we can be applying this to more data sets soon, and if you would like to see if it could work with your data, I'd be happy to show the program to you. Finally, what Sabrina wanted to do was compare 2 different parasites and see how they're recruiting mitochondria. This was great because when we arrived at this problem, we could say, well, we can just repurpose an algorithm we already have. So in this case, the user circled where Toxoplasma was and circled where Neospora was, and we found the intensity by distance for each of these parasites. And what we found was that Toxoplasma was much more strongly recruiting mitochondria. So those were my 3 projects for my first rotation. We had Metascopics, and I'm hoping in the future we can incorporate what I did in the Center for Imaging Science and CAT into Metascopics. To do that, I would be able to need to upload DICOM images, which are a 3-dimensional imaging format, and also allow for segmentation registration algorithms. To do that, I'll have to work more with WebSockets to use the algorithms that are already out there. and perhaps roll some of our own algorithms. So in the Center for Imaging Science, the big target here is just workflow smoothing. So right now it requires a lot of manual work to plug in C++, MATLAB, Amira, MEPAV. There's just a lot of things that are working independently in this workflow. And so Metascopics is perhaps a place where we could do that. And maybe there's another solution as well. So finally with CAT, I think there's a really interesting idea where instead of creating a testing procedure for our data once we have a model, is to create a method of collecting data. So before we've done any data collection, all we have is a bunch of chemicals and a bunch of tests that we're capable of doing. What we would like to be able to say is, if we have 1,000 different in vitro assays, we don't want to have to do every single assay on every single chemical just to realize later that our supervised model isn't using most of those assays. So what we would like to do is use a similar process to what we've developed for testing strategies after the model is created during model development, to be able to say, okay, first we should collect data for these assays, and then later for these assays, and then depending on what their current predictive value is, determine which assays to continue getting data in and which assays to stop getting data in. All right, thank you for listening to my Rotation 1 talk, and I'll see you soon. Oh, one more thing. Well, I would like to acknowledge the people I worked with. That's Dr. Jedinak for Applied Mathematics and Center for Imaging Science, Dr. Jane at the Center for Infection and Inflammation Imaging Research, and Dr. Hartong in the Center for Alternatives to Animal Testing Lab, along with the people I worked with in his lab, Andre Kleinzong, Vanessa Sorocha, Alexander Mertens, and finally Sabrina Nolan in the Copens Lab in molecular microbiology and immunology. Okay, thank you. Bye.